Hello, everybody. I'm Luke Timmerman, founder and editor of Timmerman Report, biotech industry newsletter. It is a real privilege and honor to be here as a visiting American biotech journalist. I've been around Boston, San Francisco, New York, San Diego, getting to know people in those hotbeds, those clusters of biotech innovation, and I'm really enjoying learning about what you have going on here. The enthusiasm and ambition in this community is really standing out. Just a few days here, but eager to learn more. Thanks to Rosie Rodriguez and the Relation team for spearheading the organization of this terrific standing room only event. So, next panel here, we have a distinguished group of folks who are trying to do a lot of the work that we were just talking about on that first panel. So very briefly, could we just do some introductions? Lindsay, would you like to go first?
Sure. Thanks, Luke. Hi, everybody. My name is Lindsay Edwards. I'm the CTO at Relation. I have an extremely mixed background. I started off life as a pop musician, as some of you know, and then ended up being an academic, and then an academic systems biologist, and then creating and running a whole bunch of different data science and machine learning functions at GSK, and then at AstraZeneca. And I've been at Relation since it was 9 people, and we're now approaching 10x that and growing rapidly.
Great. Thank you. Maureen?
Cool. Yeah, I'm Maureen Makes, VP of Engineering at Recursion. I also have an eclectic background with an environmental science degree, but really spent most of my career doing software engineering at a few different tech companies in the Bay Area and then in Salt Lake City, which led me to Recursion, which is looking at industrializing the drug discovery process.
My name is Sergei Yakneen, Chief Technology Officer at Isomorphic Labs. I did my training in computer science and math in Toronto, which was a real cradle for a lot of the modern machine learning, and so was a great place to for some of the formative years. I spent a good number of my early career working in different technology companies, but for the last 12 years or so, focusing on how technology can enable breakthroughs in medicine and in biology. Spent a lot of time working on genomics, especially in the cancer space. And then for the last 3 years have been building Isomorphic Labs, which is a machine learning focused AI drug discovery company.
My name's Nerida Scott. I head up J&J Innovation for the EMEA region. So we're a group of senior scientists and business folks who are looking for early external innovation that is strategically aligned to J&J's areas of focus, both on the innovative medicine side, but also on the medtech side. And we do partnering and collaborations as well as equity investments in companies. And we do company acceleration through our J-Labs group. So a broad sort of science business partnering activity. I also wear a couple of other hats for J&J. So I sit on the BIA board, looking at the early ecosystem here in the UK, which I think is fabulous. And I sit on the government's Life Science Council with the government's seat for innovation. So that's the industry government advisory group.
Great. Thanks, everyone. So those of you may have noticed I used the word biotech to describe what I do, and I always have for 20 plus years. But this panel is called TechBio. We're flipping this in a way. So maybe could we just start with some definitions here so that we all know what we're talking about? It's kind of a buzz phrase in certain quarters, means different things to different people at different times. So who wants to start with that? How do you define what is TechBio?
I'll go first. I mean, you're assuming we're going to agree with each other.
No, it's perfectly fine to disagree.
So, I mean, I guess for me, it's not just a gimmick. Kind of, it speaks to the fact that we're talking about companies where we start from a place of being a technology company. So we think about engineering very carefully. We think about kind of every aspect of how we build our infrastructure and everything that we do with an engineering hat on. But that doesn't change the fact that we are a drug discovery company, a relation-rate drug discovery company. So as David was saying, our reason to exist is to get medicines out of the door to patients. I think quite an important thing to note there is that those are two fundamentally different cultures. Like engineering is, engineers build things to solve problems. Scientists ask and try and answer questions. And there is an awful lot of work to do to try and get engineers to build things that can be used by scientists in order to answer questions. And I guess that's sort of fundamentally what we try and do every day.
Yeah, you don't want to be a solution in search of a problem.
Correct.
You need to build something that actually solves biological problems.
Yeah.
Maureen, I saw you nodding. How do you define TechBio?
Yeah, I think that really closely aligns with a lot of ways we talk about TechBio internally as well. And I think really thinking about the combined knowledge of those two groups sitting in rooms together, solving problems together, and our best kind of teams operate as the engineers sitting alongside the scientists, sitting alongside the data scientists, the machine learning scientists along the way, really thinking together about what's the right way to tackle this problem from each of our respective lenses and being able to draw that forward and advance that collective knowledge rather than like, this is the way we've always done it and we're just going to automate that way. Instead, how can we use that technology-enabled mindset to drive things forward?
Working together with respect for what everybody brings to the table.
Exactly.
Yeah. Sergey.
Yeah, I guess to me, TechBio is a term, but actually maybe is emblematic of a whole journey that we have taken in the industry and even my own personal journey. When I first got into the biology space, it was around when next generation sequencing started taking off. And I sort of realized a lot of data is being generated, and the scientists at the time were struggling how to actually make sense of this data, and were really like spinning their wheels. And so I initially got into it with a mindset of how can technology be in the service of science, and that was where we were at. But I think where we got to is in actually thinking in a fundamentally technological manner. Is there a real technology solution to this problem? One of the things we often talk about at Isomorphic Labs is the scalability of drug design or the lack of scalability. So can we actually design technology that means we don't have to organize ourselves into disease areas? Can we build technology systems that can reason about the entirety of the space, and how powerful would that be? And so that's what really, inspires us.
Great, thank you. Nerida?
Yeah, I might actually expand the definition from that a little bit, because I think it is fuzzy, and I think, it does mean different things to different people. And I think when I look at the universe I would define as TechBio, you could also say, you could look at it across the value chain of how do we get a product to a patient, from scientific innovation to a patient. So I can see TechBio from an engineering perspective, from a data science, AI, ML perspective, but I can also see its application being applied in target discovery, in actual product discovery, but also all the way into the clinic, into what is the tech that we use to find patients for clinical trials? How do we diagnose people? How do we combine tech that's involved in analyzing AI images? I would also say those kinds of companies are involved in the TechBio world. And when you look at that pipeline all the way through to patient real-world evidence, how do we actually find patients at risk? How do we actually try and prevent illness? How do we actually try and intercept with cures or treatments earlier so that people's outcomes are better? That along that entire spectrum, you find TechBio players. They all have a role to play. They are all in the sector of how do we use a sort of confluence of different technologies to do better medicine.
Yeah, it's a really good point that you're thinking about it from the whole vertical stack of everything that pharma does, because a lot of people have focused attention on the discovery biology aspect, which has been so hard for so long, and that's what some of our panelists here really are immersed in each day. I'll just say that in my own experience, I wrote a book about Leroy Hood, trailblazer of the genomics age, and so my own bias there is that there was a invention of the first automated DNA sequencer instruments in the mid-'80s that made the Human Genome Project possible. And at the time, a lot of people thought that was crazy and it would take 100 years with the manual methods developed by good old Fred Sanger here in the UK. No, the technology made it possible, and it was done way ahead of schedule. And now, I mean, look at how fast genomes, how fast and cheap they all are, all enabled by technology. And again, to better answer, to better elucidate some of these fundamental biology questions that people have. What's driving disease? So happen to be a believer that the revolution can come from the technological innovations first.
And in some ways, you could say maybe actually our entire industry becomes TechBio, right? Because there's no place where technology, AI data, better machine applications, machines, physical machines, as well as in silico machines, aren't going to drive and try and impact healthcare, because it costs us too much to develop a product. It takes us too long. And everything is coming back to how do we do infrastructure, how do we do data, how do we do better approaches from a technical perspective.
Can you talk a little bit about what excites you, what the big deal is here, Nerida, at a large company like J&J? Is it like trying to slay the R&D productivity dragon, the 10 years and the $2 billion and the 1 in 10 success rate for clinical candidates, that this colossus, this...
Well, when you go back to your genomics discussion, right? Actually, you look at the data of, we've typically used animal models, cell models, animal models before we go in patients, coming at problems with genomic data. And, the panel before was just talking about, 1%, 2% of your genome is transcribed into proteins. But there's so much more information coming from whole genome sequencing that's allowing us to understand human disease better at the outset. That's one way that I think is really, really exciting. It's fantastic companies here in London, in the UK, looking at genomic data, protein folding. How do you actually understand disease better to create a product for a patient that's more relevant, precision medicine? I think every step along the way, Chris said it before, we're looking for innovation outside of our four walls as any one company. Nobody can do everything. So it's really important that as an ecosystem, we understand what people are working on. Many, many nimble entrepreneurial companies here that are doing really exciting stuff. And it doesn't stop at the, can we get better at the 2 billion and get the product to approval? It's all the real world evidence and tracking people in the ecosystem, as they're undergoing treatments, wearables are they following? How do we actually manage disease better? So
there's lots of different points at which technology is intervening here. Sergei, what's the big deal for you guys at Isomorphic that the technology trigger, and then how that makes you think about what problems to solve?
Yeah, it's a great question. What really excites me is I think the technology we've been developing, it's like giving us eyes for the first time to actually observe what is going on at this very minute molecular level. And this is incredibly empowering. And a lot of the problems that we have struggled with in the field have been with our inability to actually resolve what is going on with this incredibly complex transmembrane protein. How does the binding of this particular ligand affect the conformation and actually institutes the functional change that we want to try to institute? And so building tools like AlphaFold and building out the entire portfolio of technologies that Isomorphic Labs has basically given us these eyes where we can actually resolve at an atomic level even what is going on. And the thing that I'm most excited about is then unlocking some of these really difficult challenges. So a lot of the things that we kind of dream about at Isomorphic Labs is, of course, we dream about faster and cheaper programs. That's very important, but we dream about some of the hardest targets in the space, and that's what we end up spending a lot of our time on.
Better understanding of the target gets you to faster experiments with novel small molecules, biologics, to go after those targets.
Well, the real idea behind this is rather than relying on approaches like, for example, screening massive numbers of molecules against the target and not really understanding how it might bind, how it might affect this target, actually being able to model this and model this in the computer, which means rather than waiting for 6 months for your assay to read out, you do it overnight. You schedule a job and you can then look at millions of different hypotheses, which helps you search the space. The space of kind of all drug-like molecules is something about 10 to the power of 60 large. You cannot really search that, certainly not experimentally. And so being able to do it in silico then allows you to bring that scalability. Can I run millions, billions of different hypotheses, and that will allow me to find the one that has eluded us so far for decades.
Narrow down that list, and then you go to the wet lab.
Yes.
How do you approach it, Maureen? What's the technologies that you're using? What are they enabling you to do?
Yeah, I think we've kind of had a two kind of phase approach right now in our reality, where we're generating these huge amounts of data in our wet lab up front, of this phenomics data as well as transcriptomics data and proteomics data more recently, to have this huge embedding space that can look at gene compound interactions and across the entire genome, and really starting from that broad expansive space to be able to quickly look for novel biology and find new hits. And this, we've really been focusing now through our combination earlier or late last year to be able to quickly say, okay, once we have a hit, how do we be able to use our design platform to generate as few molecules as possible to be able to get that compound that can go to the clinic? And I think the really exciting thing for us this year was looking at the CDK7 trial and their readout of they were able to, in less than a year, with around 130 something synthesized compounds, find a target from the hit ID phase. And that just really fast movement of the combined space of big data up front from the wet labs to find the areas to focus, to how do we quickly get as few molecules generated as possible to move forward, is really exciting for me.
Interesting. We'll talk about that a little more later. But Lindsay, can you say a little bit about your approach now that you've got a lot of these different types of technology tools, and we heard a little bit about it before, but what are you trying to learn from the technologies?
I mean, we're very focused on the biology of relation, because for us, that's the single most important problem to solve in drug discovery right now. If you look at clinical failures, way more than half of them are caused by poor understanding of target biology. So we feel that that's kind of where we need to spend the most of our calories. I think something actually that Maureen, in the previous answer, mentioned, which I think is really interesting, is you've got these two classes of things that you can work on. So you can work on like the faster horse. So how can I accelerate this process? How can I make something quicker? And that can also lead to transformational change. So if you think of AlphaFold as a simulator, or we've got some DNA foundation models that you can think of as simulators, it allows you to search biological space that's just intractable experimentally, because you literally have this thing in your hands where you can kind of play with it and figure out how it works. So we do a fair bit of that. And I think, as David was talking about, that's both in terms of understanding the effects of changes in DNA, particularly human DNA, but also then understanding how that plays out in the context of cells. So we do a lot of cellular experimentation as well as kind of cellular modeling to try and understand that kind of complexity of once those genes are in play, how do they interact with each other? How do they interact kind of in this incredible complexity that sits within a cell? I think what we haven't seen yet, but that machine learning is very good at, is skipping whole chunks of the process. Like if you give a model enough data of the inputs and outputs, it will solve that mapping for you. And that might be some way off, but it's worth recognizing the fact that that's where probably the huge acceleration will come. Like we can continue to build things that will accelerate bits. I think, for us actually, we think less about accelerating and less about reducing the errors and more about what new biology can we find. For me, the really exciting thing is when we see something and we know that nobody else can see this thing. We're looking at biology and it's like, no one else knows this thing.
And it starts with the samples that you collect. Now,
Yeah.
I didn't hear much talk about animal models.
No.
I heard about getting samples from patients.
Yeah.
Why is that important?
Well, because as much as my cat likes mice, I've not got a massive vested interest in curing mouse disease. It's the reason for getting out of bed in the morning is to cure human diseases, or at least make those that are suffering from them lives.
Well, but let's get specific then about osteoporosis, breakdown of bone. You're getting hip replacements.
Yep.
Those samples coming over from the hospital.
Yep.
They come to the lab, and then what happens? What do you do?
Yeah, and actually, so thank you, because we're really proud of that. Like, we have patient tissue. We do single cell on patient tissue. That data can fine-tune models. So we have kind of state-of-the-art foundation models then being trained on patient data. And then the outputs of those are tested in human functional assays. So one of the nice things about working on bone biology is Osteoblasts make bone, osteoclasts break down bone. If you want to alter how much bone is made, you need to intervene in an osteoblast. And for me, the joy of seeing that joined up thinking of like patient tissue comes through the door, single cell data goes into a model, outputs of the model go back into human cellular systems, and those predictions are tested. That kind of integration is absolutely fundamental. Let's add one small tiny thing as well. Look, it's easy to fall into the trap of thinking that we're solving machine learning problems or biology problems or drug discovery problems. But I think the real meat of this is in the intersection between those. And again, Maureen touched on this too. These are problems that we simply, I don't know anybody that has the information in their head to solve these problems on their own. So it revolves around building a culture of interdisciplinarity and building teams of people who can actually continually come back to the problem and continue to iterate across it until we get solutions.
But it seems like from the get-go, you're trying to capture the right kinds of data to feed into the machine learning algorithms, so that you don't have the garbage in, garbage out problem.
No.
Or you're just not asking, or just not capturing enough data. How do you think about, Maureen at Recursion, the types of data that you want to feed in?
Yeah, I think, we think a lot about this because we have a lot of data. But I think as far as what to feed in, for us, it's where can we get the highest dimensional signal for the lowest cost to be able to scale that up. And so that's why we do so much phenomics at Recursion is I think we feel like we could look at proteins, but that's giving us a relatively, well, we do look at proteins as well, but the level of signal is narrow as a starting spot for us. And so we originate from phenomics because we think it's a lot cheaper to run than a full animal model or human tissue in that moment. And allows us to get a really complex signal to be able to then expand from there and say, okay, what do we need to know next? What is the next piece of data that we need to bring into this system? And for us, that scale element of what can we bring up quickly to get that signal is really important.
Yeah. What data do you like to work with, Sergei?
We work mostly with molecular data for the moment, but actually I like to think of it along two dimensions, really. One is, what can we get from existing data sets? And there can be a bit of a meme out there around, well, these public data sets, they have been already mined, there's not much signal left there. And if one sort of follows the thread of the AlphaFold models that have been developed, they've all been developed on PDB. And so time and time again, you can demonstrate that with a lot of ingenuity, you can actually continue advancing your algorithms to get more and more out of your data. So there's something to be said about operating, first of all, in an environment of relative scarcity and being able to get the most out of it. But then the other dimension is, well, where can we generate really important data? And this is where, again, the interplay between models and data is really, really important, because you can actually use your models to help you predict what might be the most valuable data points that, if you were to generate those, are going to help you learn the most about where you're not currently doing great. And so we spend a lot of our time then working with our models to try to build hypotheses around, if we were to generate this data set, that will be really enabling for us to start looking at transcription factors, because it will allow us to better model interactions between protein and DNA. Or if we generate this particular data set on GPCRs, that will make that class more accessible. And so a lot of our work is around figuring out both what these data sets are and then building them, and also trying to build these data sets in a very comprehensive manner. We often talk to our pharma partners, who of course have lots and lots of data. Most of that data has been collected as part of their own programs. And so a lot of the times, this is a series of observations that are following a particular chemical series. And as that series evolves, you continue measuring different properties. And so that could be a very large data set, but it's not a particularly great data set for machine learning, because oftentimes it actually lacks the diversity that is required for generalization, which is what you often want with your machine learning model. And so, we definitely need to trade off what we think about already exist as a set of data sets and the expectations of what performance we can get, and then start generating new data sets that are comprehensive and that are tuned specifically to enable models to learn the best.
You mentioned the Protein Data Bank, that public resource. It's been combed over by a lot of people a lot of times. We know a lot about certain targets, like, say, HER2 or EGFR for cancer. Maybe not a whole lot more to mine there. But are you finding you're discovering new targets, like we're talking about for like some of these big unmet indications?
Well, our focus at the moment is less on targets, even though discovering targets is interesting. Our focus is on trying to find molecules that will actually hit some of those most difficult targets. And so the problem then isn't about how do I discover this new biology? Is it how do I design novel chemistry against this target that nobody's been able to hit because maybe it doesn't have a well-defined pocket, or it induces a very significant conformational change when it binds with your ligand, and that hasn't been amenable to existing methods. And so there's tons and tons more signal actually still in the PDB as we've been able to extract that, certainly from AlphaFold 1 to 2 to 3 and beyond.
You mentioned that you're working with molecular data right now. Are there certain types of data that you see coming down the pike that you want to work with, that you think will shed even more light?
Yeah, absolutely. And this is where I think the interest in targets really comes in. So early last year, we launched a computational biology team that we are really excited about, that will work with genomics data, that will work with clinical data, that will work with imaging data to help us start building out that biology. When we think about what does a company like Isomorphic Labs needs to do? Well, we need to build some technology. We need to run some programs that actually exercise that technology. And so when you select targets for those programs, you will often go after targets with very low biology risk, and you can take on more risk on the chemistry side, go after the really hard things. But actually, there's this whole world of yet undiscovered biology that Lindsay has been alluding to, and that's also very, very exciting. And so as we build out that biology team, we'll be looking to build models that are reasoning on top of that genetics, that RNA sequencing data, clinical imaging, to help us better understand the stack of phenotypes, all the way from kind of molecular phenotypes to cellular, tissue level, organism level, or even how we interact with the environment, because a lot of the disease is tied up in these different layers.
So starting with this technology-driven and engineering mindset, kind of laying down the basic fundamentals for discovery of new biology. I know that we talked about this the other day, paying attention to trends and developments in other sectors like technology. And I think Tesla and Waymo provide a good example of how you go about the question of developing self-driving cars by capturing different kinds of data and training your algorithms on that. And Tesla, for those not familiar, many cars on the road for many years, capturing lots and lots of visual images and feeding them into an enormous database that it is using to teach its algorithms how to drive cars by themselves. Waymo took a different approach and doesn't have this giant fleet, but did small pilot projects in carefully defined areas and gave it a whole lot more, even with cameras and radar and LiDAR, a very, very rich set of data, and then also a whole bunch of synthetic simulations that they're running around it. And they actually leaped ahead. I was just in San Francisco, as David was, and rode around in the Waymo, and it worked really well. So it's interesting, like there's two different ways that they've shown to get to the same point, and Waymo's ahead. Do you find anything instructive in there and how you think about capturing the data and giving the algorithms something to really chew on?
I mean, there's this kind of machine learning 101 relationship between the types of models you can train, types of questions you can answer, and what data you have access to. I mean, the easiest way into it for me is like the model can't understand what it can't see, and maybe that's the moral of the Waymo story. Like it might have to have a huge amount of visual data to be able to learn about things that would be much easier to learn about if it gets them from some different modality. And the same is definitely true of biology. So you're looking at these awesomely complex systems. And I think it's a bit naive to think that if you look at one slice like RNA, you're going to get all of the information that you need. In fact, I'm sure that there are cases where you see cells that look exactly the same through that lens, but they will respond differently when we perturb them. And so the model is limited in what it can learn. So I think it's a really instructive analogy. And it's just a question of trying to figure out what are the right lenses to look through, what's the minimal number of lenses to look through to get you where you need to go. And also, like really critically, how do you scale to the kind of scale that we need? Because possibly outside of recursion, there's not a huge number of data sets in the world that are at the scale and of the quality that's required for the sort of things we've been talking about today.
Maureen, I know your folks think about this a lot too. What types of data? What do we need to be looking at?
Yeah, I think when you talk about the models can only know what they can see, I think that really resonates. And I think that's one of the areas that we've been looking at is how do we know what's missing in our data sets? And then how do we really think intelligently about generating the data that we're missing, particularly on the chemistry side of the world, where there's kind of more to explore for us right now is what are the ways to train our models to tell us that information is important? And so I think that I've lost my train of thought entirely. I'm going to point to Sergei.
Yeah, what do you think, Sergei?
Yeah, I mean, maybe if I was to say something about the Waymo Tesla analogy you've brought up, is I'm not sure it holds here. I guess the way I'm thinking about this is to solve these problems, we're going to have to throw the kitchen sink at most of these. You know, science, both exhilaratingly and kind of unnervingly, doesn't work in a straight line way where you say, this is my target, I'm just going to generate data until I hit that target. You may get there, but then the data set size would be really astronomical and not realistic. And what ends up happening, especially in machine learning, is it's just a very empirical science. So you're going to have to try a lot of things. You're going to have to generate data, different data sets. You're going to have to burn a lot of hardware. And you're going to get there with one approach or another. But I wouldn't say that there's one that's like, let's just generate massive data sets and everything will fall in place. I'm not sure that will get us there.
Can I ask Sergei a quick question?
Yeah.
Oh, sorry, Nerida, you go.
Well, I was just going to say, I think the fact that you have different models and different approaches, different scale, means when you're partnering as well, that people have different philosophies on what they will place a bet on and how they will peel the onion, right? You know, what's the equivalent of the MTA on the TechBio side, right? Looks very different. You know, if you're, focused on small molecule versus, somebody else is looking at a different molecule, whether it's a large molecule or other modalities. I think that's also a really interesting aspect of this space, which is really, really immature. And people are exploring how they partner, how they share risk, and how you balance the interests that folks have in a very novel kind of set of ways. And you see disruption, right? We saw disruption this week, right? It is a space where some surprising bets might play off and they might look very much cheaper or might have done something very much faster. And then you'll see an aggregation towards that.
So Nerida, from the buy side of this table, what would really make you sit up and say, I'm impressed?
So for a company that is, going all the way to the patient, a lot of what's going on is incredibly fascinating and it's incredibly powerful if it works. The question is, how do you know what's going to work? And you'll see, investigations that come out with chemistry that you would never have got to with traditional methods. But you have to still see, does that work? Does that create the efficacy? Does it avoid, for example, in oncology, does it avoid the resistant strains because it's been designed, in at the beginning by applying the model with all the different biology that is tracking out of iterative, oncology states. So a lot of it for us is like, how do you pick the winners? How do you guess some? What do you wait and watch and see? And how do you actually do it? Because there's a lot of stuff going on internally at large companies as well. And how do you do that sort of partnership? It's not one solution.
Proof will be in the data. Show me.
Yeah.
Okay. I know we're almost out of time, so real quick, I just want to ask people, lightning round answer about the London's Knowledge Quarter, which we're sitting in. Do we have all the pieces here? Or maybe what's missing? What would you like to see?
Yeah, I'm happy to go first. I think London has a fantastic set of things going for it. In terms of TechBio, the UK has fantastic other tech as well, fintech and all sorts of links. So the idea that you can have a confluence of skills, I think, is really strong here. We have London Life Sciences Week, which will hopefully outstrip JP Morgan as an event.
Ooh, bold.
And we have TechBio as a group. And, I know we're going to talk about the ecosystem in a panel next, but I think there's fantastic crossover of world-leading biology and tech here.
What do we have here and what's missing?
I mean, so I was born here, so I think it's an amazing city. I've lived elsewhere, and so I've had the joy of coming back to London. It's one of the great cities. It really is, and it's got everything. So, I mean, that's important. And the fact that we've talked about the conflation of, 3 or 4 of the great universities within a relatively short distance of here. So it's all here.
It's a magnet for talent from around the world.
Yeah. Luke, you've been everywhere. So you're visiting at the moment. What do you think? Like, how does London feel to you as a place?
Well, I wasn't ready for that. I am, like I said, I'm listening, I'm learning. I think I am an American, so I have my bias. I believe in the American.
Okay, so the weather and the
spirit.
transport are terrible, but no, but I mean, how does it feel compared to somewhere like Kendall Square?
Kendall Square is unique on Earth.
Yeah.
There is just a beehive of intellectual energy like no other place, like on a per square foot basis. Yeah. So that is something to look at, and but I actually see some of the fundamental pieces here for sure with the great universities and a base of technology talent, a world-class city that attracts talented young people from around the world, definitely, and even some large pharma. Placing some large investments. So I think a lot of the pieces are in place and coming together, a supportive local political community. I wouldn't be surprised if 20 years from now this looks a lot more like Kendall Square.
Amazing.
Can I have a take?
Yeah.
I guess my take on this is, before being in bio, I used to work at Amazon for a little while, and one simple sort of heuristic they had is, go where the talent is and bring yourself there. And I think London's got that in spades, with all of the different colleges. And so the talent is definitely here. But then that talent wants to take risks. That's what entrepreneurship is all about. And I feel like there could be more capital. There could be more appetite for risk taking where, if I'm having this conversation with folks here, a lot of the times somebody's working in their startup, or they've been between academia and startups, but most of the time it's like one startup. And when I'm having this conversation with somebody at Kendall Square or at JPM or in the Valley, people are on their 5th startup. And so I think there's just a lot more appetite to take on risk, try an idea, see where it goes. If it doesn't go anywhere, there's no stigma associated to that. You just go on, you try your next thing. And I feel like we haven't quite got here yet, even though maybe that appetite is increasing, but I'd love to see more of that.
Culture is super, super important. I say this all the time when people ask about China too, why I'm still bullish about America. But last thing, Maureen, got the last word. I think you're the youngest person here on the panel. You like living here in London?
I do. I mean, I moved here 9 months ago to open our office in King's Cross, and I think I've been really blown away by just the support from the rest of the community and people coming to our event and inviting us to their events and really welcoming us in. And there's sort of been this mentality that I've experienced of everyone really cheering for each other. Like, there are no lack of diseases to cure right now. And I hope all of our hypotheses from respective ways are right and that they can all be, so we can all go after that. And I think people have given me recommendations on restaurants and running groups and all of this, and just like, let's make this happen together. And I think that's been really positive, but could use more sunshine.
All right, with that, please everyone thank our panel.